All right, I think we're going to get started. So, hello everyone. Thanks so much for coming to our trainee rounds today. I'm Julie. I'm one of the T-CAIREM Education Trainee Affiliates, and I'll be adjudicating today's session. The Temerity Center for AI Research and Education in Medicine at the University of Toronto has been made possible by the generous donation of the Temerity family. T-CAIREM is an interdepartmental center that serves as a focal point for collaboration amongst healthcare providers, trainees, researchers, computer scientists, engineers, and industry. Our goal is to transform health through AI. A quick note to the attendees for today's session, please use the chat function in Zoom to submit your questions for the presenters, and they'll be addressed during the discussion period at the end of each presentation. By attending this event, you're also consenting to being photographed or recorded for non-commercial use by T-CAIREM. Today's first presenter is Reza Basiri. Reza is a PhD candidate with the Institute of Biomedical Engineering at the University of Toronto, and his project is titled Advancing DFU Management, Introducing WoundVista 2.0 for Enhanced AI-Powered Diagnosis and Education.
Hello, everyone, and thank you, Julie, for the kind introduction. Actually, the title of today's presentation is slightly different, but same topic, still same topic. Okay, here we go. Okay, so I'm grateful to be here presenting my work at this venue. So the topic of the presentation is synthesizing diabetic foot ulcer images with diffusion model. As mentioned, I'm a PhD student candidate at Biomedical Engineering, and my supervisors are Dr. Milish Popovich and Dr. Sheraz Khan. And my work is a collaboration between Institute of Biomedical Engineering, CAIT Toronto Rehabilitation Institute, and the Zivot Limb Preservation Center in Calgary. So a brief introduction of the center. CAIT located at 550 University Ave, just next to Mount Sinai, is a research center focused on rehabilitation. So basically, we focus on rehabilitation of patients with spinal cord injury, stroke, and elderly population who have difficulty with mobilities and whatnot. In my research, I focus on diabetic foot ulcer. Diabetic foot ulcer is a skin condition, is a skin wound, basically occurring mostly at the bottom of the foot on bony structure, on bony areas. The trigger factors for diabetic foot ulcer are vascular insufficiencies, which is mostly stem from the diabetic condition, and excess skin pressure, stress, moisture. And the prevalence of diabetic foot ulcer among the diabetic population globally is 6.3%, and in North America about 13%. And majority of the patients are elderlies, above 65 years of old. And the risk of not treating DFUs are first, loss of mobility and independence, hospitalization. Patients with diabetic foot ulcer are 3.4 times more likely to be hospitalized, and amputation. So out of all the amputations done, 85% are related to diabetic foot ulcers. And so the patients with diabetic foot ulcer and above 80 are 5 times more likely to have amputation. Diabetic foot ulcer, it's a condition. Basically, it's a multi-element condition that it could be stemmed from one of the 3 primary pathways, either by damage to the nervous system, peripheral neuropathy or autonomic neuropathy, or through peripheral vascular diseases, which limits the blood delivery to the area. So all of these pathways basically are triggered by diabetes. So in summary, diabetic foot ulcer is a condition that requires, when analyzing and investigating different aspects of the diabetic foot ulcer, we require to look at it from different angles and include a comprehensive kind of a data set if you want to actually have a global view of the condition. The treatments for diabetic foot ulcer basically is a cycle of monitoring, where physicians basically monitor the area, the depth, the visual cues of the diabetic foot ulcer, prescribing therapies to encourage the healing process, and routine change of dressings and primary care. So in my work, basically, I focus on the monitoring aspect and a little bit to the therapy aspect of the diabetic foot ulcer treatment. So the current monitoring processes are done mostly manually by tracing the area of the wound, using rulers to measure the wound size, or a bit more advanced ones where they use imaging to basically capture image and then draw the surrounding area on a computer rather than on a paper. So because as I mentioned, diabetic foot ulcer is a condition that requires a comprehensive view of different elements that are contributing to this condition. We looked around and we couldn't find actually a data set, a wholesome data set that basically include all different aspects that we are interested in investigating when we want to investigate how to go about treating diabetic foot ulcer, or how to increase the effectiveness of the current existing treatments. So to remedy this issue, we decided to create our own data set. A data set that we investigated literatures, and then we determined the significant descriptors, significant elements that are important to analyze. We didn't know exactly, or for 100% certainty, which are like the more significant or less significant. We basically did a global search and then compiled all different possible elements that should be considered in a diabetic foot ulcer analysis dataset. So here we basically built this small imaging apparatus in this small box, where we have a thermal camera and a depth camera to synchronously take image of the wound using these two image modalities. And in addition, we developed a protocol for collecting diabetic foot ulcer data set, metadata, and demographic data. We compiled this case report form where we included all different important aspects of this condition, or the elements that contribute to this condition. For example, smoking habits, alcohol consumption, if there are any history of heart conditions, cancer, are there any toe conditions or arch conditions, and so on and so forth. So basically, we create this standard protocol for our data set, and also encourage the future data collections in diabetic foot ulcer to use this standard method of data collection. So with collaboration with Zivot limb preservation in clinic, we were able to start this data collection of about 300 patients. We intentionally selected this particular clinic in Calgary because this is the only clinic in Canada, a multidisciplinary clinic in Canada, where all the different types of specialties are gathered in one center. Whether that's vascular surgeons, podiatrics, infectious disease teams, endocrinologists, and whatnot. all gathered in this one center to basically focus on vascular diseases and in particular foot ulcers and diabetic foot ulcers. So we figured this is a really good basically center for us to collect this comprehensive data set where we are looking at different aspects of the disease. So once we collected this data set, we called it Zivot data set to show our gratitude to the clinic where they help us to create this large reference data set. We conducted different, usage of the data set where we developed a deep learning model to detect diabetic foot ulcers in images. So as you can see on the left, these are examples of our model basically picking up the diabetic foot ulcers in the image. We were able to also increase the specificity of the model where you see on the bottom right in image F, there is a wound, not diabetic foot ulcer wound on the basically above the ankle, which this model was perfectly able to categorize it as not diabetic and didn't draw basically the square around it. But like everything else, as you can see in the image, has been picked up by the model. So this basically helps with the process of monitoring. As I explained earlier, my work is basically focused on enhancing the monitoring aspect of the diabetic foot ulcer and increasing the effectiveness of that step, basically. So also another application of this dataset we have deployed is to classify the stage of the healing of the wound. So in the protocol that we designed, we also ask clinicians to label the stage of the healing, whether it's inflammatory, proliferatory, or remodeling. And then we develop a machine learning platform based on the images and the metadata collected, which is, temperature, as I explained, smoking habits and all these sort of things. The machine learning model is basically able to predict which phase of the healing that one is currently experiencing. And from there, because a lot of therapeutic modalities, they require this information before they can be prescribed to the patient. So by using this basically machine learning platform, we are able to create customized or wound-specific therapies as opposed to generic therapies that are currently being prescribed due to lack of resources. So we noticed the importance of diabetic foot ulcer images in analyzing and basically treatment of diabetic foot ulcers. The two important aspects of the images in this area are first, images are mainly used to train clinicians. Not every clinician is expert in diabetic foot ulcer. So they need to go through trainings. They need to basically process or learn by visually looking at different kind of diabetic foot ulcer images. And so images in that sense is very important for training. And also we noticed that there are other basically data sets out there that either small or they're not very comprehensive. So by employing a way of enhancing or augmenting the existing data set, we would be able to basically create customized data set. We would be able to create a larger customized data set for future AI researches in this area. So we decided to, as I mentioned, we decided to make the data collection or creating data set in this area easier, because by creating synthetic data set, we would be able to rectify the data imbalance. We would be able to create customized data sets, which, it would be rectifying the imbalances that we see in demographics of the existing data sets. We would be able to reduce the data set cost collection. and all the other difficulties that goes with the dataset collection, ethics and all the times that goes into it. And also we would be able to provide a dynamic way of medical training as opposed to presenting one image or a few collection of the images over and over. In tests and in medical trainings, the institutions would be able to have like a dynamic kind of a presentation of different kind of conditions. So we deployed diffusion models. First, we used GANs, and then we moved on to diffusion. Basically, diffusion is the process where an original image is fed into the model, and gradually noise is added to the image, where at each step, we try to go back to the original image by using a unit, basically, architecture, as shown at the bottom of this figure. So by doing so, we are able to basically learn how we can generate the diabetic foot ulcers. The model can learn how to basically, what are the elements of diabetic foot ulcers, and then by mix and match, basically by diffusion, different kind of elements, the model is able to create new cases of diabetic foot ulcer. Here's an example of visually how the diffusion model works. Basically, we have the original image at Z0, and then gradually random noise is added to the image. At the end, we have like purely random noise. But at each step, basically, we try using the unit to go back to the previous step or to the original image. So this way, this basically the unit architecture at each step learns how to remove noise from any given image or get any basically noisy image to drive back the original image. So we set up a diffusion model particularly for generating diabetic foot ulcer as explained on the specifics of the model here. And then we also use FID, KID for quantitative evaluation. And also we use 3 podiatric surgeon evaluators to basically go over our generated images versus real images to rate them whether they are real or fake. So we gradually improved our basically ability to generate synthetic data set. I'm going to skip this one. So first, we had a fixed set of 50 basically synthetic images and 50 real images. At first, we focused on only the wound area. So we basically cropped out all the other parts of the image. We focused on zoomed in on the wound only. And in a very fixed manner, we ask physicians to basically rate these real or fake from 3 physicians. And here we can see the results where on the left is the rating of the physicians on real images, and on the right is the rating on the synthetic images. Where 0 means that nobody was able to rate them correctly. 1 means that one physician was able to rate that particular image correctly, 2, 3, and so on and so forth. So you can see the real images, basically, physicians are more confident about rating them correctly. And on the synthetic images, they are less confident. But the average rating, basically, 2.52 and 2.1, they are within the same range. They're not significantly different. So this proof of concept basically showed us that we are, in fact, able to generate new synthetic diabetic foot ulcer images to basically reach our 2 goals. So we gradually increase the quality of the generated images, and also we increase the scale of the evaluations we do. So we created this Wound Vista platform, the online platform. As if you want to visit it, you can scan this barcode here, QR code here, and or visit the link mentioned at the bottom of the image, woundvista.com, where basically anybody could go and start rating the images, whether they are real or fake. Of course, we have separate ratings where the rating comes from clinicians or general public. So here in this one, we generated more images rather than 50. We had about like 100, 200 images where images are randomly displayed, and then the users are basically able to rate them real or fake. As of now, the real images and fake images are within about the same, I guess, percentage of marking, 55 to 46, average of 51. But then we are continuously improving the wound vista platform. We are increasing the quality of the images generated. Here is a quantitative measures that we deployed on the generated images. The take-home message here is that 70% of the times, clinicians mark synthetic DFU images as real DFUs, which is a very good starting point. Here is the latest of the generated images. These are fake wound images generated. So we are increasing the resolution of the images. And also we are adding the ability to have prompts where the users would be able to basically specify the kind of the wound they require or they want to have as an image. Whether they want to have it on a dark tone skin, whether they want to have it on the heel or on the metatarsal areas or on the hallux or whatnot. So there are a few limitations with with diffusion models. There is basically a privacy concern whether the diffusion models can retain the information of the original images. So these are, the gray areas that are being explored and then needs to be addressed in more detail. And so for the future step, basically we are enhancing, or as I said, we are continuously enhancing the wound vista quality of the generated images. We are also adding on other options to basically have prompts specifying the images. We are enhancing the prompts that we can also use to generate images. Thank you so much.
All right. Thank you so much, Reza, for your presentation. That was excellent. So now we'll open the floor to questions. First questions from the adjudicators. So Dr. Ma, do you have any questions for Reza?
Yeah, thanks for your, can you hear me?
Yes, yeah.
Okay, perfect. Yeah, thanks for your great presentation. Really nice work. My name is Jima. Currently, I'm a machine learning lead at UH AI Hub. Basically, data sets are the foundation to build machine learning models. I believe your data set and the data synthetic method will have high impact in the community. So I have some technical questions. So you used the latent diffuse model, right?
Yes.
And the image size is 256 by 256.
The input, yes. The images are larger, but we downsize them to 56 to 56.
Okay.
Yeah.
So basically, the original latent diffuse model is designed for the high resolution image generation. For the input image with a size of 256 by 256, actually the original diffuse model can already handle it. For example, the DDPM. So did you get a chance to compare with the latent diffuse model with the original DDPM?
Yeah, so we use the stable, we started, as I mentioned, like we started with stable diffusion and then latent diffusion. So the thing is that diabetic foot ulcer images, the feet basically are not very consistent. There are amputations, there are amputees, and each individual foot looks differently. There is no, consistently among them. So the only part that basically is consistent is actually the wound area. And that's mainly why we only focus on the wound for our preliminary generation of the images. So yes, we use the latent diffusion with the standard latent diffusion, but we had difficulty with generating the toes and like different amputations that exist in this population.
I see. I see.
Yeah, so and the data set size, as I mentioned, we have 270, about 300 patients. So this, which is about 2,000 images. So 2,000 images may be enough to basically have the model learn the specifics or the semantics of the wound, because the wounds are basically consistent, but the variety in the foot deformities is so much large, that it's vast, that this size is basically wasn't able to have the model learn, the toes and like the deformities that exist in this population.
Okay, yeah, makes sense. I also noticed that for the noise scheduler in the model, the time step was 1 sound. This is a typical, this is a typical setting during training. But during inference or during sampling, if we still use 1 sound time step, it would be too slow. So my question is, did you use any faster sampling strategy during inference?
Yeah, so the inference, it could be anything less than 1,000, and so we use the smaller time steps for the inference to save time, but I guess to enhance, like to basically employ the full power of the model, or to generate the highest possible quality, we opted to use the 1,000 steps.
Okay, yeah, okay. So last question is about the evaluation. So in general, we hope to use the semantic data to boost the downstream task performance, for example, segmentation, classification, detection. So did you get a chance to augment your original real data set with the semantic data set to see where it can boost the downstream task performance?
No, this is undergoing. So for the classification, detection, all these sort of things, it's an undergoing project to integrate it with the original data set in order to investigate the enhancements of the the methods.
Okay, I see.
Yep.
All right, and I also have just a quick question, since we do have a bit more time. I'm wondering, with wound detection and also with anything that kind of has to do with skin, there's a lot of concerns with AI models and bias, right? And being able to identify the same conditions on different skin tones. Have you done any work to take a look at how well the model generates images that reflect different depths of skin tone?
In terms of the generation of the synthetic images, I'm aware of that, and no, we haven't looked into the variety of the skin tones. That's a very valid question, and it's a very interesting area to look into. And that's actually one of the, I think, the main application or main benefits of being able to customize wound generation images, is to hopefully at the end we would be able to specify the skin tone, so we would be able to minimize the imbalances that exist in the data sets.
All right, thank you so much for your answer. We're going to move on to the next presentation now. Our second presentation today is by Elly Zhou. She's a PhD candidate with the Institute of Medical Science at the University of Toronto. Her presentation is titled Developing a Digital Lung, a Deep Learning Approach to Simulating Physiological Lung Function During Ex Vivo Lung Perfusion.
Okay, thank you so much for the introduction, and I hope everyone can hear me okay and also see my slides okay. Okay, so thank you all for attending the session today. My name is Elly. I'm a second-year PhD student under the Department of Institute of Medical Science, and I am currently co-supervised by Dr. Shafka Shavji and Dr. Andrew Sage. So today I'll be presenting you my PhD project titled Creating Digital Twins of Human Lungs Using Ex Vivo Lung Perfusion and Machine Learning. So I just want to spend a brief moment talking about ex vivo lung perfusion. So EVLP is an organ reconditioning platform that keeps lungs perfused, stabilized, protected when it's outside the human body. And the circuit is composed of a few key components. So first, we have this closed loop circuit to keep the lungs perfused through its vasculature with a cell-free solution. And then we also have an ICU-grade ventilator connected to the trachea that keeps the lungs breathing at a specific rate. And we also have a deoxygenator to remove oxygen from the circuit, as the lungs will be the oxygenator itself in the circuit. And in addition, there's also a temperature control unit that keeps everything at a physiological temperature, which is 37 degrees. And given its unique features and abilities, EVLP has really been used as a therapeutic platform to study isolated human lungs. In addition to the fact that currently EVLP runs 6 hours clinically, but our previous studies have shown EVLP can be extended to 72 hours or up to a week. So this really further enabled our group to test a wide variety of therapeutic strategies, including gene therapy, stem cell therapy, and also how to build universal blood type organs. But when you think about the conventional clinical study design, there are a number of challenges we need to consider. So first, when comparing the control and treatment arms, the large variability within the data would cause results interpretation to be significantly challenging. And secondly, the scarcity of human lungs will limit the availability of the lungs we can use for research testing, as this is very different from using, for example, cell cultures or animal models. And additionally, this is also very costly. Each EVLP alone would cost around $50,000. So now the question is, how can we use machine learning to address these challenges? And before really getting to the topic of machine learning, I just want to spend a brief moment here to highlight that EVLP as a therapeutic platform can really capture this clean set of data of the isolated human lungs without systemic effects of the human body. And the data is both organ-specific and multimodal. So in terms of data modality, we're looking at image data, transcriptomic, proteomics, metabolomics, lung physiolomics data together with the clinical history of the patient. And all these data can then be combined with multimodal machine learning to create a digital twin of the human lung. So talking about the digital twin, what is it exactly? A digital twin is like a virtual version of something real. It is a digital copy of a physical object, system, or process. And this copy would allow us to simulate, analyze, and understand how the real thing behaves without having to interact with it directly. For example, the digital twin of the cars have been widely used to monitor and optimize fuel consumption, to predict engine failures, or plan for future maintenance. And not only in the engineering field, digital twin is also an emerging concept in medicine. For example, to build a digital twin of a patient. A digital twin of a patient is a virtual model that represents a person's physical and physiological characteristics. And people have proposed to use it in personalized medicine, predictive healthcare, and also to monitor chronic conditions such as diabetes or heart disease in order to make real-time adjustment and treatment plans. And the concept of this digital twin really is getting more and more attention in the medical field recently. So here's a paper published in early March this year, and this is a review paper describing the process of developing human body digital twin using 5 steps. And the main idea here is that as we include different data modalities into the model, the digital twin gets more complex and capable. For example, the level 2 model is a predictive model for forecasting future health conditions by incorporating past data of the patient. And as we incorporate more data such as treatment record, the level 3 model considers human interventions such as drug treatment or gene editing to analyze and predict the health outcomes after the intervention. And soon after that, another paper came out with more of a focus on the application aspect of digital twin in medicine. And one of the areas highlighted in the paper is the development of novel therapeutics. Because currently, clinical trials typically involve the use of animals and patient cohorts. But with the digital twin model, it will be possible to reduce the number of animals used in preclinical trials and also to optimize patient trials using virtual patients. For example, we can screen large number of drug targets and drug candidates, and the doses and combinations can also be tested by the medical digital twin model before giving it to patients on a personalized level. And I hope by now I have convinced everyone that digital twin is a great idea in medicine. But the question is, given that it's so amazing, why aren't we there yet? The answer really comes down to two parts. First, human bodies are more complex than something a machine could ever predict, because we simply just don't behave like a machine or like a robot. And the second point is that we need to really find the sweet spot between having any data set versus having a high quality data set on non-diseased subjects and also disease-specific subjects. So coming back to our EVLP platform, it really generates this valuable data set on non-diseased lungs, and also it provides opportunity for us to study disease-specific lungs. So having said that, the first objective of our study is to develop a digital twin of a human lung, which is a complete suite of forecasting models that predict human lung function using multimodal ex vivo data. So by combining with multimodal machine learning, we aim to develop a model combining tabular data, high-resolution time series data, X-ray image, and microarray data. And the second objective is to validate the clinical utility of the digital twin approach in therapeutic development by doing validation using real-world data. And summarized here is a full panel of parameters divided into 5 categories. And under lung physiomics, we can see parameters like airway pressures, compliance, and oxygenation. And under metabolomics, we have lactate and glucose. For image data, for example, we have X-ray images and its derived features. And for the transcriptomics data, we are looking at the gene set enrichment score for specific genetic pathways. And for the model, the patient age, sex, and also, for example, total lung capacity were also considered. So altogether, we're really looking at digital twins of human lungs comprised of over 150 multimodal algorithms, and each one of them has been specifically optimized for the training setup for each target feature. And moving on to methods, the study population included clinical EVLP cases from 2008 to 2024 at Toronto General Hospital, and different data modalities were processed and modeled differently here. So for the high resolution time series data, we used a gated recurrent unit model to do the time series forecasting. And for the tabular image and microarray data, we used an XGBoost regressor model to forecast its behavior in the future. All combined together, we're using MAE and MAPE as model evaluation metrics. MAE measures the distance between the observed and the forecasted values, and MAPE measures the same thing, but just expressed as a percentage of the observed value. And here we also have established 2 modeling strategies for digital lungs, the static and the dynamic. So first, at the start of a case, we have baseline data that shows basically how the lung is doing during the first hour, for example. And with the static digital lung approach, we use baseline data as input, forecast onto the second hour first, and then use the real baseline data plus the forecasted second hour data to predict the lung behavior later on. And as for the dynamic digital lung, the model is being continuously updated with the most recently available data. So here you can see that the model gets continuous update from the second hour data and then forecast to later time points. So with that in mind, I'm going to show you the results on the model development and performance. So here we're showing digital twins of human lungs can accurately forecast a full panel of lung physiology parameters. The static digital lung accurately forecasts, for example, mean airway pressure within 6.7% of the observed value, the arterial partial pressure of oxygen level within 8.3% of the observed value. And also the dynamic digital lung also accurately forecasts the same list of parameters, but with better accuracy. Because as the model has been continuously updated with the most recent data, this is going to make the prediction more accurate. And here, moving on to the high-resolution time series data. So over here, we're showing that by using this high-resolution data, we can really generate a more accurate digital twin. And the idea here is that for a subset of physiological parameters, including compliance, mean airway pressure, peak airway pressure, and expiratory volume, we have per breath high-resolution data, meaning that instead of having one data point per hour, we now have a value measured for every breath. So from the table, we can see that the GRU model and also for both the static and dynamic trained on this data showed improvement in forecasting performance for all parameters as compared to tabular data forecasting. And in terms of lung biochemistry, the digital lung model also showed good performance in forecasting a list of biochemical parameters with 1 to 12% accuracy, depending on individual parameters. And also the dynamic digital lung also showed improved performance. And in fact, for a lot of parameters, the MAPEs here were improved by around 50%. And as for the image data for a given X-ray, we first derived top 10 principal components using the CNN-derived latent features. Combined with lung function during EVLP, our goal is to forecast the image PCs of the third-hour X-ray image. So our previous studies have shown that these image PCs were strongly correlated with clinical radiology features, such as consolidation or pneumonia. And we wanted to see if the correlation between the radiology features and the forecasted PC values still hold true. So as you can see in the table here, for the all 4 radiology labels that are correlated with the observed PC data, the correlation is still significant for the digital lung forecasted PCs for both the static and dynamic approaches. And another very interesting finding is that the digital lung model accurately forecasts transcriptomic changes in human lungs. So here we have the lung tissue sample microarray data. By passing it into GSEA, we can obtain the enrichment score for various pathways. And using the enrichment scores as input, together with the lung function data collected during EVLP, we aim to forecast how the pathway would change in the future. So as shown in the table, digital lungs can forecast transcriptomic changes within 3% of the observed enrichment score value for lung disease-related pathways. And they're all listed in the table here. Just a few examples, such as hypoxia, TNF-alpha signaling pathway, and also inflammatory response. So by now, we have seen that the digital lungs can accurately forecast a full panel of parameters related to lung physiology, biochemistry, image, and transcriptome. So now the question I want to ask now is, do digital lungs have clinical utility in the real world? So now I'm going to present you the results on digital lungs in a clinical setting. So here we have a drug called alteplase, which is essentially the tissue type plasminogen activator, also known as TPA. So TPA has thrombolytic effects and is given under clinical suspicion of pulmonary embolism, which is shown in the 2 images here. And the picture on the right is showing you a blood clot in the lung. And TPA is usually administered to the EVLP perfusate around the end of the first hour of perfusion to break down blood clots. And it is known to reduce pulmonary vascular resistance of the lungs. And given the protocol of TPA treatment during EVLP, so we want to know, can we validate the therapeutic efficacy of alteplase using digital lung? So here we're showing the comparison of parameter values between suitable lungs and unsuitable lungs using both the conventional approach and digital lung approach. So under the conventional approach, we're comparing 2 independent groups, whereas under the digital lung approach, we're using a paired analysis approach for both the suitable and unsuitable lungs. So the idea here is that the digital lung model would predict lung function as if no treatment was given. In other words, the digital lung serves as the control cohort of the study, and the observed data would serve as the group with the treatment. In this case, it would be TPA. So this really has allowed us to use the paired analyses, because now we have the same lung, and the digital lungs generate the data that's as if it's untreated, and whereas the observed values represent data when given the drug effect. So if we look at PVR here, which is the parameter that's expected to change under TPA treatment, we did not observe a significant effect. But given PVR is a calculated variable, we also looked at pulmonary arterial pressure, PAP, which is one of the parameters used to calculate PVR values. And we did see a significant difference in the suitable lung group. This is showing us that digital lung can enable detection of drug effects that would have otherwise been missed. And additionally, the figure on the left side is showing one of the suitable lungs. So you can see that the digital lung forecasted pulmonary arterial pressure values, which is in orange, are higher than the observed values. So this is showing that this is indeed in the same direction as what we expected for a case that's been treated with TPA. So in summary, we have shown that multimodal machine learning models powered by EVLP data can accurately forecast lung physiology, biochemistry, image, and transcriptome characteristics. And second is that digital lungs provide a framework towards using digital twins to validate therapeutic efficacy. And in terms of next step, we're conducting proteomics analysis for digital lung model, and we would also like to incorporate additional proof of benefit experiments of the digital lung model using other clinical therapeutics. And a big thank you to my supervisors, Dr. Andrew Sage and Dr. Shafka Shavji, my committee members, Dr. Bo Wing and Dr. Amy Madani, for their valuable guidance in the project. And I would also like to extend my gratitude to all the colleagues who have supported the study. And thank you very much for listening.
All right, thank you so much for your excellent presentation. So once again, we'll do some questions, beginning again with the adjudicators. So Dr. Ma, do you have any questions?
Yeah. Hi, Valley. Thanks for your nice presentation. Really great work. I like your slides. It looks very visually appealing, and as well as your results, especially for the real world evaluation on the drug effect. So again, I have some more technical questions. So for the ventilator data, how do you handle the potential noise and artifacts in this kind of time series data?
That's a good question. So we developed an entire pipeline to basically clinically annotate each breath. So we have 9 clinical labels to determine if a breath belongs to which category. And one of the 9 labels is noise. So we have been able to use that label to filter out the noises.
I see. Okay, makes sense. And so for the second question, many studies have shown that synthetic data can be used to boost the model performance. So in your task, the training set is actually not very huge. So could you please comment the idea of using synthetic data to enhance your training set, just like Reza have presented?
That's a good question. So we actually did some experiment with the GRU model training, because for that training, we only had 170 cases, which is already a lot. But then we noticed that by adding the static features, the model performance actually improved when it comes to like forecasting the next hour time series data for the breath parameters. So yeah, static feature does help. And we currently are adding probably 10 to 20 static features along with the time series data into the GRU model to like fully boost up the performance.
Okay, yeah, sounds great. Yeah, that are all my questions. Maybe we can leave some time to the audience for asking questions.
Yeah, for sure. If anyone has any questions, please feel free to put them in the chat. Dr. Ma, I mean, if you have another question, we definitely do have time to take it. So please feel free.
Okay, yeah, I have one more question. So, for your framework, you have a separated image classifier to extract the image features first, and then compute the PCA components, and after that you incorporate this the PCA components to your next framework. My question is, did you try do you try to build an end-to-end pipeline to directly incorporate both the image and the time series features in an end-to-end framework in your future work?
I see. That's definitely our next goal in the future. But we haven't had the time or chance to. But that's a good point. Yeah, I think all the pipelines should be combined at the end of the day, to take all the input all together and then classify them into different groups and different modeling, and then generate the output together for sure. Yeah.
Yeah. Okay. Thanks.
All right, and I do have one question for you as well. I know that you brought up this great example of alteplays, and I'm wondering if you have started to think about other clinical scenarios in which you want to validate these virtual lungs, and specifically how those scenarios will kind of push the boundaries of the physiology of those lungs in different ways.
Yeah, I think the answer is absolutely. Because one of the focus for the lung transplant research is to find a way to repair and treat the donor organs before they go into the recipient's body. So I think if I stay here to talk about like different therapies that we can validate the digital model on, I can probably talk for an hour, because our group has been spending the past, I don't know, decades to test different validation therapies. So I think the answer is definitely yes, but here I'm just showing you, the digital lung can be used as a framework to test autoplays, for example, as a starting point.
Okay, yeah, thank you for the response. I guess also what I'm kind of getting at here is like, what aspects of the physiology of digital lung are like the most kind of, that doesn't have the hardest time with, the ones that you really want to validate because you want to push the boundaries of what the digital lung can do, the aspects of human physiology that they don't quite capture well yet. Do you have a sense of that?
Sorry, could you, sorry, let me just make sure that I understand your question right. Are you talking about the physiological parameters that we can forecast using the model? Or you're talking about the entire human body?
Sorry, yes, the former, just because, different clinical situations require the model to be good at forecasting and having an understanding of different physiological parameters, right? So what things is it kind of not as good at?
Oh, I see. So currently, our model can take in 24 different physiological features. But as we continue our analysis, taking in protein data, taking in high resolution pressure data, or even we can get into the territory of single cell analysis, right? Like we can basically map out the entire human physiology as we have more data that comes available. I think that's definitely one of the future goals, but that it takes time.
I understand.
Good question. Right. Good thinking.
Thanks so much for your presentation. Thank you to both of our speakers for their excellent presentations, and to everyone for attending today's session. This is the final trainee round session that we're holding this summer. We do have videos of the previous 4 trainee round sessions on the T-CAIREM website at tcairem.utoronto.ca, so please do check those out. And also, before we sign off, a quick final reminder to our summer students who are attending today's rounds that the final mini conference is on August 8th. So if you're a summer student, just make sure to put that in your calendar. Thanks everyone again, and have a great afternoon.